Sure.
Yeah, the one I hit was the one Joey said. Did you flip that? There you go. That still be the front, yeah. We can do that. It's still the same thing. These little lights are outside then. All on our own. You don't work? No, they're broken. They're not. This is it, actually. I think that's okay, yeah. Okay.
Okay, so I'm just going to get started since everybody's settled down and everything. So my name is David Bridwell, and I'm a research scientist here at MRN. So the title of my talk is Human Brain Dynamics Above and Below the Scalp. And since I have like hometown advantage for this talk, and since this is related to engineering, I wanted to talk about my background and how I got into EEG, which involves a lot of people who also have background in engineering. So before I started grad school, I was working on this project called the Shama Project. So it was one of the most intensive studies of meditation training conducted to date. In terms of the number of measures they used, there's 12 different tasks with EEG collected during most of them, as well as blood measures of telomerase and cortisol and facial expressions and physiology. And also in terms of the amount of time that the individual spent meditating from pre to post, it was the most. So we have this retreat center in the Rocky Mountains, and it's glamorized in this photo by all the snow and everything. And the BBC came and they were going to take photos, which was very cool, except they never used them. And I learned an important lesson because you had to take a day off work to be an actor or whatever in their thing, which nobody wanted to do because we worked a lot at that time. And so I did that. They were going to pay me a day's salary, but they never did. So I learned if you're going to get paid for something, you have to get the money up front. And then this is the guy I work for, Cliff Sarin, pictured with the Dalai Lama. And I also learned that if anybody you know ever meets the Dalai Lama, they will tell you about it, and everybody gets very excited about that. So my job was to run, set up the EEG labs and help collect EEG on all the participants. So here is our system. Everybody at the time, it was UC Davis Center for Mind and Brain. Everybody was very excited about the biosemi systems with these active electrodes. Because back in the days, you had to scrape the scalp a little bit to get a good recording, and you didn't have to do that anymore. So I never had to experience the abrasion that some people had to experience with EEG. And at the time, we considered this portable EEG. It was portable, so here's the device along with the battery pack. It's portable in the sense that the participant could walk up and pick it up and walk out the room with it. And it was pretty easy to ship to the retreat center. But it's much different nowadays, where these are portable devices, which I'll show you the results for in our experiments. But this, if you purchase one, you get this very fancy briefcase here. And there's 4 mobile EEG devices within this briefcase. So the ones that the studies I showed you are this small for just 4 electrodes. That's the active wave, which is depicted there. And then we've got 2 of these smarting devices, M-Brain Train with 24 channels. So they're very small now and much more portable than what we used to consider portable. So people are very interested in using, given the portable EEG nowadays, people are very interested in measuring EEG while people perform natural tasks. So we did an experiment where people watch movies, and others are doing experiments with multiple people hooked up at the same time, and they record their interactions. So that's called hyperscanning. And then we do things like we stare awkwardly into each other's eyes, right here, measure EEG at the same time. That was our photo we took yesterday. And so we have the 2 systems, and we're trying to think of some experiments to do for hyperscanning. So if you know of anybody undergrad student who is interested in being one of the, in helping with that, then let me know. There's my email. So while I was there, when I had time off, basically you didn't have much to do, so you would go find a rock somewhere and read a book. And this was the best place to read. So the retreat center was over there, and you'd hike up all the way to the rock. This is the Great Stupa of Dharmakaya, which liberates upon seeing. It's right there. So this book is very good, Rhythms of the Brain. And Buzaki says, I've learned as much about the brain by reading about novel branches of physics and engineering, math, and computer science as I did from studying papers directly dealing with nervous tissue. This is like, this makes everybody in the room very happy right now. This is the crowd to make that statement to. And I also read this book. It's by Ramesh Srinivasan and Paul Nunez, and then I went on to grad school with Ramesh. And so let's get into EEG, the basics of it for a little bit. The first thing is that it's actually amazing that you could take somebody with proper IRB consent and put electrodes on them and be measuring their brain responses within a few minutes. That's an amazing thing, and when Hans Berger first discovered it in the 1920s, he didn't believe it at first, and he had to do a lot of verification to make sure it was actually true. So the EEG responses arise from radially oriented pyramidal cells with synchronous postsynaptic potentials. And so they arise, and then they have to pass through the skull-scalp boundary where they're recorded by an EEG electrode. And then they're going to return back to the brain in a return current, so they're for current conservation. And here you can see this is a simulation of brain potentials as they pass through each of the layers here. And the main attenuation occurs at the skull. And then at the skull-scalp boundary, there's a lack of conductivity, which reduces the potential over the source and spreads the voltage tangentially across the scalp. So in some ways, that's a filter. Well, it is a filtering process. It's spatial filtering, but it also temporally filters. So it ends up emphasizing lower frequency EEG oscillations. And in the book, they said, nature has conveniently provided us with an anti-aliasing spatial filter in the form of a poorly conducting skull. So they say it's a good thing, which if you're really into EEG, you'll find a good way to view everything. But one of the things it means is that the question of how many electrodes do you have to put on the scalp to get adequate coverage. There's devices that go all the way up to 256, but because of the spatial smearing, you don't really need 256. You're okay with 128, for example. And then if you go up to these type of devices where we've got 8 channels, then you don't really get adequate spatial coverage. So you're undersampling, and it's a Nyquist issue, just like the temporal Nyquist issue that we are familiar with. So in those type of cases, you don't want to plot a topographic map of the EEG because it's misleading and you didn't accurately capture it. But people don't seem to do that, which is very nice. So in the book, he talks about, this is where you're measuring with EEG. It's as if you're watching an orchestra, and with ECOG or local field potentials or single unit recordings, you would imagine that you're putting a microphone on an individual within that orchestra. So you're getting a very specific picture of that individual. But with EEG, you're capturing this global, large-scale thing that's happening, and it more represents the sort of musicality and combination of whatever that orchestra is producing. So that's the thing that when you listen to it, you enjoy, and it's great. So there's benefits to that in the sense that when you space average and temporally average the signal by the scalp, you're emphasizing things that unfold around timescales of our perceptual experience. And you're capturing these global things that are aligned with our cognitive states and things like that. So this is what Edelman and Tononi said. So as the spatial grain of conscious experience is coarser than that of individual neurons, the temporal grain appears to be coarser than that of individual spikes. So our conscious life unfolds in seconds. So the search for neurocorrelates of consciousness experience should focus on neural dynamics that unfold on similar timescales. So that's the importance of the timescale here. And it's important in this perspective to notice that EEG is not like an impoverished view of single units or ECOG. It's not just the ECOG is the great thing, but then you put this thing on top of it and it screws it up or anything. EEG provides its own measure of this own unique thing, which you can't necessarily get, a picture that you can't necessarily get with those other measures. And it emphasizes timescales of cognition and perception, and as a consequence, it also tells you a lot about the state of the brain. So here he says, evoked potential activity may reveal more about the state than about the physical attributes. And this is, when I was getting into grad school, there was a lot of early studies that focused on vision. And then I realized this influence of attention in your state, and seeing that attention was the appropriate way to go because that's sort of the global measure that the EEG is capturing. So if you ask an engineer, the best way to understand a system, the brain is a complex system. How do you understand it? You have to provide an input, and you're going to measure outputs. So to an engineer, well, these are some different ways. There's frequency tagging, which is what I'll talk about, presenting natural stimuli, presenting discrete events, and then averaging over those events for a ERP. So with the brain being a complex system, you provide an input, measure the output, and make inferences based on the relationships. So literally, when I say that what an engineer would say, how do we understand this complex system? This is a book, Random Data by Bendat and Pearsall, which is very good. They say the most straightforward empirical approach is to subject the system to a sinusoidal input and measure the output and amplitude and phase as the input frequency is varied. So this is what I'm depicting here in this frequency tagging study. You present an input as a fixed frequency, and then that goes into the brain. There's your input frequency, and then you measure the output electrode response within that frequency. So that's the application of this approach. And then Basar has a lot of great stuff, and he's not really talked about as much probably as he should be. And here's one. So according to general systems theory, all information concerning the frequency characteristics of a linear system is contained in the transient response and vice versa. So knowledge of the response allows one to predict how the system would react to different stimulation frequencies if the stimulating input were sinusoidally modulated. So in the context of this experiment we did, imagine your auditory cortex. It's always doing something, right? It's always active to some degree, but you can place a 40 hertz auditory impulse through your ears. It sounds like a helicopter. And that will drive the auditory system, or so that when you place an EEG electrode, you can measure an EEG response at 40 hertz. So that you know what that response means. You're providing this input, and that response is literally driven by that 40 hertz input. And it simplifies your representations of what's happening. So then you can do various attentional manipulations, and the interpretation is relatively straightforward from there. So in that sense, when you present a sinusoidal input, or square wave input, I guess in this case, it's acting like a filter to filter out a certain aspect of brain activity. Here it's 40 hertz auditory system. And different systems, different networks have different preferred frequencies. So with the auditory system, you do a really good job with 40 hertz inputs. And with the experiments I did as a grad student, 8 and 12 hertz around the alpha range, visual inputs was very good for driving frequencies that were related to attention. But you can try to get into these different networks with these different preferred frequencies. And when your input frequency matches the network's frequency, then the responses are enhanced, like resonance. It's literally resonance. So with the Tetris study, basically you just provide the auditory inputs. It's the same across all experimental manipulations. And then you either have people attend to those auditory inputs while they stare straight ahead at the failing Tetris screen, or they play a very slow version of Tetris or a very fast version. So the idea is if your auditory system is going to be able to process this input to the same degree when you're attending to that input or when you're engaging in these complex visuospatial tasks that could draw upon your resources. And this is sort of a measure of like, what are you getting with this measure? You know, what is it providing for you? And for us here, it provides a measure of the degree in which you're attending to this difficult visuospatial task. So it's a degree in which you're attending away from auditory information in some sense. So here, when the Tetris task is difficult for everyone except one of the subjects, the responses are reduced. And then when the task gets easier, these 40 hertz auditory responses increase. And then attending is a very small effect. And the responses are related to the difficulties. So those who had the most difficult task we found the most reduction in the 40 hertz response. So carrying that further, like what are these frequency tag responses revealing about our perception? That's what I showed here. So here we have this flickering texture. So it's going at 8 or 12 hertz, 8 or 12 times a second. And then people do tasks at the attended location. So the response I'm plotting here is to the exact same physical stimulus. It's always this 8 or 12 hertz flicker. So the question is, and then when we vary the task, the question we look at is how does that response reflect whether you subsequently correctly detect a target within the task or if you miss a task. So for each of these 3 tasks, separately, we look at whether they correctly detected it or whether they incorrectly detected it. In this task, higher responses to the flicker are related to correctly detecting the target. But in another task, higher responses are related to failing to detect the target. And the reason why is because the flicker feature aligns with the target features in this task. So in the pre-stimulus interval before the target, if you're representing the target feature well, this flicker is coming in with those same features into your visual system, and it's also getting enhanced in its representation, which means when the target appears, you do a good job of seeing it. In this condition, the flicker feature competes with the target at the attended location. So when you're representing that target feature, you're not doing a good job of being able to detect the target that's going to appear. So it's associated with incorrectly detecting target as opposed to correctly detecting targets. So there's some really neat stuff you could do with frequency tagging in terms of individuals' perceptions and attention. So this was Javier Garcia's work, where he showed a various range of flickering textures, kind of like this one, or flickering orientation gratings, like this. And then by considering the responses across all of them, he devised this forward encoding model just like that from Mariah, where you can measure attentional response across the orientations. And when they attended to the orientation, you found a tuning curve that aligned with the attended orientation versus when they didn't attend, they didn't find that. So there's this forward encoding tuning functions, which is very interesting. And then in this study, I measured attentional tuning functions. So, yeah, I mean, this idea of tuning functions with EEG is straight from the very first perspective seems like you can't do that, but with this approach, you can. So basically, you measure the response to this flicker in every single condition. So every response I measure is to the exact same physical stimulus. You just adjust which so the stimulus has an orientation. You just adjust the orientation that the individual attends to monotonically. And that response, so when you have to detect a target with that orientation, or that's 10 degrees, 20 degrees off, the response declines. So this is basically like a tuning, so it could be symmetric, just like that, except it's a very complex level of tuning, right? Because the inputs come in through your orientation cells in the brain, and they propagate up to these higher areas which are involved in all these complex things in the global networks of EEG. But they fall off when the orientation is offset. So it reflects a different type of tuning function, but it's focused on attention. And we showed that it differs by different tasks, and the individual differs across individuals, and individuals who use an optimal strategy in a particular task have better behavioral performance. So so that's the approach of frequency tagging, which is very nice. I like it a lot, and basically for its interpretability of the responses that you see, it's very nice. And in some sense, we'll think about natural stimuli now. There's a decent overlap between the two. It turns out with frequency tagging, you can understand your input just by Fourier analysis of your input and Fourier analysis of your output. And that's because your input has that frequency structure, just the periodicity. With natural stimuli, you can't simplify your inputs like that, but you have some complex input that depends on how you look at what, in the context of a movie clip, what you think is a relevant thing. You could look at a pixel and say this is how the pixel is modulated, but the brain's probably not looking at a pixel. Or you could look at faces and say this is when a face came on and things like that. But it's very difficult. You have to make the choice of what's the relevant feature. The alternative is to measure responses across individuals to the same inputs, the same movie clips, and then look at the correspondence across individuals. So that's inter-subject correlations. And the idea is you've got a complex input. You don't know what the output of the brain will be necessarily. It could be nonlinear, and it'll be a summation of different evoked potentials to the different aspects of the stimuli. But that doesn't matter as long as that nonlinear complex thing is similar across individuals. Or at least with this analysis, when you correlate responses across individuals to the same complex input, you're emphasizing those complex responses that are similar across them. So the main thing we wanted to look at is what information is retrieved from these cortical responses to complex stimuli. So the first is, what can you tell what stimuli they watched? Can you tell what movie clips they watched? The second thing is, are these responses related to their preference for the movie clips? So we had them watch 12 clips, and they vary in their, there's some romance, there's comedy and drama, and awkward social interaction is like one of my favorite things. For some reason, I think it drives a lot of brain responses, but there was like 4 of those. So here's the, we had a very simple way of predicting which movie clip they watch, based on correlating across all subjects and then picking the one with the highest correlation. And across the 16 clips, this is chance right here, 6.25%. We were above chance for each clip. For Silver Linings Playbook, we got up to 52.9%. And I think it varies based on, one of the factors is the content of the clip. So those that tended to be more dramatic tended to have higher assignment accuracy, and the comedic ones tended to do not as well. So that seems to be one of the important factors. But there's indication that you can begin to predict which input they're experiencing here, and then looking at their clip preference. I thought this was a very elegant way, a very simple way, which I guess those are kind of related, elegant and simple, of looking at whether their preferences were related, movie preferences were related to their EEG responses. So they rated each of the 16 movies from 1 to 5 based on how much they liked it or disliked it. And we looked at the correlation among two individuals in terms of their rating. So that would be the Spearman's row. And then their intersubject correlation. So those that have high similarity in the degree in which they rated the clips, or in the way that they rated the clips, if they have high similarity in EEG responses, then their ISCs would be high. So they would sit around here or like this, but we don't see that's the case. So basically, they're unrelated. These cortical responses are unrelated to clip preference. So we got to keep looking and thinking about what these responses, what information these responses can provide us. Or it could be related to your subsequent memory of the clips. Or like I alluded to here, it could be related broadly to the clip content. There was a study that did, I think it was scenes from The Walking Dead, where they looked at people's responses in Twitter posts, and the parts where people were most engaged corresponded to the most Twitter posts about the episode. But it wasn't necessary, but that was stronger than the individual's own reported engagement. So it's this sort of like broad level of engagement that not necessarily each person is aware of or able to report. Things like that are something to think about. And so the next thing is discrete events, which is the most common way of analyzing EEG, where you present these discrete inputs and then collect an ensemble of responses and then get the ensemble average. So that's a ERP right here, and you can see the peaks, and they've assigned names to the peaks. And so here's what happens when you flash up a visual input to a monkey. You can see this is over 115 milliseconds. It comes into the visual cortex, begins to spread to other areas. In about 65 milliseconds, there's a lot of the brain that's already responded to this input, and so on. So there's a lot that happens even within the first 100 milliseconds of flashing up a stimulus. And when you're getting over here into 80 milliseconds and such, that it's not depicted in this figure, but the frontal responses are going to feed back into the occipital areas. So there's this constant feed-forward, feedback, circularity loop going on in the brain, which you can think about with EEG. It's less apparent with fMRI, where over 1 or 2 seconds you're averaging the response based on your acquisition as well as the temporal filtering of the hemodynamic response. But even when the visual input initially first enters the occipital area of the brain, it's already affected by the whole brain, right? That area is always in communication with every other area. So that's always happening. And I like how Uzaki says it in his book. So he says there's no real structural top in neuronal hierarchy, since the higher order information in the brain can be immediately forwarded back down to other areas. And then neuronal information is propelled in parallel in multiple juxtaposed, superimposed loops, making the distinction between top and bottom processing very difficult. And I really like these things, because there's a tendency to say there's a top and there's a bottom and to think along those terms, but we want to remember how complex and how quickly information travels across the brain. And the idea of synchronicity gets at that. And it's really emphasized with EEG. And then he goes even to more extreme to say synchronicity corresponds to an acausal connecting principle as opposed to causality, which is the modern prejudice of the West. So you have to be a little bit dramatic when you write a book and make quotes and things. So the thing with ERPs is that you end up summarizing them by their latency or their peak. And in Bassar's book, I like this. I thought this was kind of funny, because he notes here that the average evoked response, the ERP, which I showed you before with all the peaks, is described in terms of these arbitrarily defined components, latencies and magnitudes. These arbitrarily defined components, they depend on the location of recording electrode, behavioral state, or sleep stage and upon the nature of the stimulating signal, and he should add reference electrode to that list as well. And then goes on to say again, interpretation of it. So he says arbitrarily defined components 3 times within this paragraph, which is what I find funny. And I think the concern is that the ERP is an average, so each single trial is this input with these cascade of oscillations that superimpose. And when you average across the individual trials, that average isn't necessarily the thing that you see on an individual trial, and it further superimposes these different things. And then we assign interpretations to these peaks, like awareness of errors. And probably it's not going to be the case that each individual peak is going to have an individual interpretation, like awareness of errors and things like that. It's probably a bit more complex than that. So when people think about this, for me, independent component analysis kind of deals with that, because you take that superimposed oscillations from your input and you decompose it into these independent temporal responses. So this is decomposition of an ERP, and there's different sources that represent different aspects of the peaks, and then you can analyze those sources. So to me, that attenuates the sort of maybe fuzziness that happens after all the averaging and then labeling process. And the other thing, like James Kavanaugh and Mike Cohen, when they come and give talks, they talk about wavelets, and they really emphasize that. And that's a great way to get a time frequency representation of individual trials, so that you're also decomposing the response into the frequency characteristics where EEG happens. So there's distinct frequency bands. You can try to isolate those. So I was very happy to get to say this in this paper, that because I was dealing with the issue of labeling the peak. This is a PE, or then we got into discussions with Vaughn, who was here at the time, Vaughn Steele. I go into his office and he'd say P3B and P3A and P3 and these things, and my brain started to gloss over with all the terminology. And so I was glad to say here that the PE, which is technically the name of, so it's very robust. If you put in an input, you see a response between 200 and 500 milliseconds, which we call the P3. There's a lot of inputs you could put in to find that, and but typically it's emphasized the most in oddball tasks where you present something unexpected. So they call it the PE. If you analyze responses to errors, and the PE indicates it's a response to errors as opposed to P3, which might not be. But there's a tendency to think that because we gave it a different name, it's a different thing. So I was happy to say the P3 and PE should not be considered distinct due to their distinct titles, however, as they appear to overlap to some degree in their neural significance and psychological attributes. So there's studies out there that show the similarity between them. And the thing is, in general, when I do these studies, I try not to depend on assigning a name and assigning a function to each peak. And usually, you don't always have to. So Steve Luck has done a great job of talking about this in his ERP book, the issue of what you have to do if you want to think that it's that component in order to draw on all the previous studies that have said it was that component and given it these interpretations. And it overlaps a lot with the reverse inference issue and fMRI, which Russ Poldrack talks a lot. So it's something to think about. But I'm happy that I never really felt a strong pressure to label the peaks in my studies. And you know, the thing is, if you find that this peak is associated with a certain cognitive function that's supposed to be related to your task, that's information right there. It means that the group that had an impairment in this peak were impaired in that cognitive task, and you know the timing of that impairment. You know it didn't appear at this thing that happened at 100 or 150, it happened at 200 milliseconds. So you've got details on the timing of these deficits related to your experimental task. And I just thought I'd throw this out there in the Squires paper with Steve Hilliard, where they first published on the PA and the P3A and P3B. They say in the paper, we do not intend for the labels P3A and P3B to become institutionalized and only use them as a shorthand notion for the present paper. Indeed, at this time, there are much uncertainty regarding how many different types of P3 waves can be properly distinguished. So this was the first paper, and this, I think, probably stopped after the first paper. You know, people started to just give them the titles and go along with it. But we do acknowledge the complexity still. So we're doing these discrete inputs, and this is the typical task here. It's an oddball task where you're presenting tones at a certain frequency that are more common than tones that you're presenting at a different frequency. So these are called the oddballs. And the thing is that the brain is constantly trying to understand and represent the environment. It's always doing that work, and it always has predictions about what it expects. So that's its primary function. And so if you're sitting in your office, for example, you know if there's people around you, so you create an expectation somebody might call your name. If you don't think anybody's there and somebody calls your name, that would freak you out. And that's an indication that you made a prediction and the prediction was wrong. And so Rodolfo Linus talks about this. His book is very good, where he talks about basically every perception in all our processing is geared towards interacting with the environment. Like that's our function, is how are we going to manipulate and move about in the environment. So they're very intertwined. And even if you just have somebody sit there and listen to these tones, these systems will get engaged. So you can get a measure of these systems that are involved in understanding their environment. Just with a simple task like this. But there's some added complexities. So typically you'll average the responses to each of these oddballs, average them together as if they're the same level of unexpectancy. But here, this is using Edie and Baldi's surprise model, where you take account of the history of the sequence of stimuli that precede each input and consider how unexpected it is. So people create a probability of how likely each of the two are, and then they update that probability with a new input, and the difference between them is how unexpected it is. So if you have to update your probability a lot, it means it was more unexpected than if you didn't have to. So if you organize the stimuli based on that metric, you see that this is more unexpected or more surprising, this is less, and this is less. And according to this measure, if you have 3 of these rare appear, the next standard is actually more surprising than some of these rare targets that preceded. So it's a more complex way of looking at the unexpectancy of the targets of the stimuli. And here, so typically these responses are analyzed by number of preceding non-targets. So here, if you plot the number of preceding non-targets and the ERP amplitude, there's a nice period from 0 to 5 where it increases, and then it sort of flatlines. So around 10, if you've got 10 non-targets, or the unexpectancy of a target appearing after that is about the same as if you have 20 non-targets. So it doesn't matter as much anymore at that point. So if you're analyzing your data based on number of preceding non-targets, you might not be capturing all of the information. So because we use, for example, with correlation, right, you expect a linear relationship. That might be here from 0 to 5, but it might taper off. And if you look at the surprise of each of the targets, you see that surprise follows a similar pattern as the actual ERP amplitudes, where it tapers off as well. So now if you organize the ERP amplitudes by surprise instead of by number of preceding non-targets, you can see that the relationship becomes much more linear when we use our linear statistical methods, you have a better chance of detecting that relationship. So this is a process of representing environmental stimuli, and we looked at impairments in schizophrenia that could be related to issues with not being able to process these sensory regularities in your environment. Because the symptoms of schizophrenia, they're very complex. Things like attention and memory and these complex behaviors can emerge, these deficits can emerge from deficits at very low levels. If you don't have a nice sensory representation of your inputs, then it's going to be very hard to create appropriate outputs. So here with healthy controls, this is their sensitivity to surprise. And within schizophrenia, you can see that it's attenuated. So basically we showed that healthy controls are more sensitive to the surprise of the targets, or the unexpectancy of the targets, or prediction error, 3 different terms for it if you want. So individual patients with schizophrenia were less sensitive than healthy controls. So they demonstrate cortical differences in context processing, 340 milliseconds following rare targets. So this is the source, temporal source that we measured amplitudes to. So we can tell at what time those deficits appear, right? And I don't care what it's called, this peak, but we know it's related to your sensitivity to the statistical regularity of events in the environment. And that's saying a lot. So it's consistent with impairments in working memory, fragmented perceptual experience in schizophrenia. So there's studies that looked at preceding non-targets and shown deficits in schizophrenia. This just shows it in a different way, which is looking at surprise, the surprise metric. But we're only also analyzing responses to targets here. So to see the differences between healthy controls and schizophrenia, we didn't even have to look at non-targets, only targets, which is it's another sort of additional measure that you can use to look at that. And then also the group temporal ICA, which is what I used for the previous study. I used in this study. It just showed that these are responses to a non-target when the person accidentally presses a button. They're supposed to withhold, but they accidentally press. And the idea is that when you do that, there are a bunch of processes engaged in the brain which are related to correcting your error. It's realizing you made an error, making a correction, and various cascade of events that happen. So, there's a series of sources, a series of evoked potentials, which likely represent different aspects of that cascade of events. And looking at this group, which there was a wide range of depression, self-reported depression among this group. Individuals who reported greater somatic symptoms of depression, which is like lethargy and things like that, have a reduced ERP response to their behavioral errors. So something in their processing of the areas of errors is compromised in these individuals that report higher somatic depression symptoms. But we don't see that with affective symptoms. So this is like irritability and things like that. So the thing here is that we're combining cognitive measures from your ERP responses to this cognitive task, along with self-reported clinical measures of depression. We're trying to look at the relationships that exist between the two. So that's what we showed here. So it's very useful. I found that this temporal ICA is very useful for providing the different peaks, decomposing the ERP into different peaks, which you can analyze and generate interpretable results. So I'm very happy with it. And then I don't have too much time, so there's the idea of rest as well, resting state. And we extended that group temporal ICA. We're also using it on resting EEG, and it's also worked very nice. So as part of that, we evaluated different algorithms, different decomposition algorithms to see how they behave. and generated this way of generating realistic simulated EEG from simulated wavelet coefficients, and then interpolate that across the scalp so that your simulated sources are very biologically plausible with these widespread responses and frequency characteristics typical of EEG from like 0 to 4, 4 to 8, and so on. So the idea is your observation of the spectral information of EEG, take 2 seconds, take the spectral information from 0 to 32 hertz. There's a mixture of these underlying sources, and with group spatial spectral ICA, you can decompose that. So there's a lot of algorithms in GIFT, and some algorithms are better than others. So I found in terms of stability and correlation with the original source and the derived source, in the simulated data, we found some recommendations. Combi worked out very well, and then it depends on the source characteristics in general, which ones may work better than others. But if you look at real EEG data, these are the type of sources you expect. So with Combi, each of these 10 here were decomposed. Also, Infomax ICA decomposed each of these 10, and Fast ICA did pretty well. So Combi, Infomax ICA, Fast ICA, they're not getting at the same thing. So potentially you could do these different algorithms to isolate different subsets of these sources and see what information you could get complementary from it. But yeah, the sources are very nice. Here's the same analysis, group spatial spectral ICA with EEG data collected concurrent with fMRI. And the sources have frequency characteristics similar that you'd expect from EEG. There's a number of alpha ones, which is nice because alpha, which is 8 to 12 hertz, is thought to be many different alpha sources within the brain. So we found a few of them, and then with this approach, you get a individual subject EEG time series, the mixing matrix from the spatial spectral group ICA, which matches with the mixing matrix of fMRI. And you can look at the relationship by deconvolutions, which assumes your fMRI response is, at one point, is related to your EEG response at a previous point. Generate an impulse response function, which should resemble the hemodynamic response function, and then find that, for example, alpha was negatively related with a bunch of occipital regions, but then delta theta and some of the beta gamma region, fMRI, beta gamma EEG is positively related to a subset of these fMRI regions. So this is better spatial spectral characterization of these EEG responses by combining the two. Yeah, so I talked about some different approaches for EEG and some different interpretations of things you could do with it. And with frequency tagging, the oscillating visual or auditory inputs were presented, which entrain different cortical networks by their different input frequencies and reveal distinct properties related to perception or your visuospatial engagement. With ERPs, you could decompose that response with temporal ICA. And we showed in 2 cases that the ERP amplitudes are related to attention and cognitive control with patients with schizophrenia and a population with depression. And then that multi-subject decomposition we use with spatiospectral analysis of EEG, which could be resting EEG. It has considerable utility with generating interpretal spatiospectral maps, and we were able to link them with concurrent fMRI for better spatiospectral characterization of these brain dynamics. So thanks to the co-authors and to MRN and funding sources, and thanks for listening.
So I was thinking about the study on predicting the responses to videos. And you mentioned that the person itself is anticipating something, and it's different, and that causes humor. It's different from what they anticipated. So, do you
yeah.
think that complicates the problem?
I don't know, in terms of humor generating an unpredictable response, you'd expect it would do a better job, or at least that aspect of humor, I think, would better contribute to predicting those movie clips, because that's one of the most robust EEG responses is unexpected. I'm not sure how unexpected humor is, and then I'm not sure the engagement level. I guess the basic interpretation is I think the engagement to comedy is reduced compared to drama, where it's a lot more complicated.
It's sort of a...
People get, you don't want to be exposed to something that's going to be difficult to process or make you feel a negative way, which dramatic things can do that, but with comedy, you're in a safe space.
A lot of work has been done on using EEG for detection of deception. In fact, there's a product that measures the P300, and the claim is that if the subject recognizes the object, there'll be a very big difference than the object not recognized. So if you show the person who wants to get a visa an AK-47, and a person says, I don't know what that is, but there's a strong P300, then that Pakistani lady is not going to get a visa. What's your reaction to that? It's used.
Yeah, there was a study where they looked at trying to detect threats, and the idea was if there's a threat hidden in the image, there will be a P300. I mean, yeah, it seems like basically that's one of the strengths of EEG, would be detecting those cases, and so it seems reasonable given that you have enough single trial resolution. And that's one of the things about the temporal ICA, like with the surprise thing in schizophrenia, you're basically doing a single trial analysis, so it helps to do that ICA pre-processing and really capture the response. So if you combine it with some advanced processing of the EEG, that could be helpful. I mean, I don't know if you're asking about the ethicalness of it, which...
No, the real technical issue is, can a person who is a good liar somehow mask this effect?
Okay. Yeah. I'm going to say that they possibly could, but I
There's
don't
lots of
think...
countermeasures.
Countermeasure. Can a person use a countermeasure to cover this up?
Yeah, when you lie, you typically have more, that's one of the studies was you have more frontal engagement because you have to construct a story. I don't know how EEG, that was an fMRI finding.
J. Peter Rosenthal, the guy who's publishing these articles on countermeasure proof, EEG detection of lies, but it's all lab-based, it's all guilty knowledge task-based. No one's taken this to the real world, demonstrated sensitivity and specificity, and published it. People have done varied numbers of those things, but no one's done
No,
that
real
all
world.
once.
The one thing I remember reading the real world is it tends to be useful for detecting deception in males. It almost never works for females. Figure that one out.
Yeah. Any more questions? Yeah.
Maybe I can make sure clear just related to the talk. So the difference is like in novelty or you expect it or not expect it, is that isn't the result you showed is like an average, average how different it is. Can that reliably detect in a single trial or single person?
I mean, I won't do as well. There will be a lot of noise. There's a lot of noise in single trial EEG. So I don't know what you mean by reliably detect, because
Like, you feel P300 is a word, actually word job, like people saying something which is commonly they say every time. And then, so that this effect can be detected just by one person in the trial, or you have average group effect size versus individual effect size?
No, I mean, like within each individual, you can look at their sensitivity to surprise, because you have individual single trial amplitudes across the range of stimuli presented, and you'll see, that there's a linear structure to that. So at least at that level, you can get each individual's sensitivity to surprise.
How big is the inter-individual difference? For one person, this antidote P3 is high. For other person, the same antidote P3 may be considered low than possible.
I mean, it's pretty similar across individuals. In general, you'll see it at the individual level. So, yeah.
This is old stuff now by now. I haven't read the literature for a long time, but I seem to remember there being some old studies of heritability of P3 amplitude and how sort of reliable it was. I don't know if anybody can speak to that more recently. That's stuff from the 90s.
I don't know, it'd be interesting. Like, what does that mean? It could be related to.
Well, I guess just with respect to what she was saying, that sort of sort of individual differences in people's sensitivity to surprise. It seems like there's within subjects reliability there, but definitely individual differences somehow.
Yeah, there's an
Big
attention.
enough that people are looking for heritability rates.
Yeah, there's an attentional factor. So there could be there's probably many mediating complex things that are also heritable that relate to attentional things. So yeah, it's interesting. ERPs are related to neurotransmitters, so you can try to get some measures of that. There's a lot of interesting stuff. Okay, cool, thank you.